Hello, welcome back to my channel by Shansi. In today's lesson, we are going to look at two things. We are going to look at the significance of telomeres in the process of mitosis. We are also going to look at stem cells. So let's get started right away. Please don't forget to subscribe to our channel. When you subscribe, you show me that you appreciate the work that I do. You are providing me with support. So I appreciate when you subscribe and you share with your friends. And then I also appreciate when you give me a like. So let's start with the significance of telomeres. But before you look at the significance of telomeres, what are telomeres? In our previous lesson, we were able to learn about the structure of a chromosome. And we saw that a chromosome has telomeres on its tips. So these ends of a chromosome are called telomeres. These ends of a chromosome are known as telomeres. So what is the significance of those telomeres? Or what are telomeres? So when you look at the tip of this chromosome, this region known as a telomere is part of DNA. However, it is made up of short base sequences that are repeated many times. What does that mean? It means that you could find that this region has nucleotides that are just repeated, maybe T, 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 then C, 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 then G. That is all it is made up of. These nucleotides, or the nucleotide sequence, does not code for any proteins, but they are really very important, as we are going to see. So telomeres are made up of DNA with short base sequences that are repeated many times. Telomeres have one major role. Their role is to just make DNA a bit longer. What does that mean? Let me use this illustration to try to show you what I mean. Assuming this is our DNA molecule, this blue region is the DNA molecule. So usually, the enzyme that is responsible for duplicating or replicating DNA, it does not copy the whole DNA molecule. Assuming this is the enzyme will come and copy just this section, leaving some ends of the DNA to just copy just this section, leaving some ends of the DNA. So that is the enzyme DNA polymerase. It stops somewhere before the end of the DNA molecule. What does that mean? It means that this information that has not been copied is going to be lost. This information is going to be lost. So that is why God created telomeres. So assuming these are our telomeres, the telomeres are going to lengthen. They make the DNA longer. So the DNA is going to be made longer, such that if the copying enzyme comes in, then it will be able to copy all the important DNA. So assuming the copying enzyme comes in, then it will be able to copy all this information from this length to this length, including the telomeres, which are not coding for any protein, but we shall also have all the important genetic information, all the genes copied. So that is what this statement means, that telomeres make DNA a bit longer, allowing the copying enzyme to complete copying all the meaningful DNA. Assuming this blue section is our meaningful DNA, it means that when the enzyme that copies comes in, to be copied maybe from this section to this section, meaning that all the meaningful DNA is going to be copied. Now, this bit that has been lost would be just part of a telomere, which was not coding for any protein, so no important information will be lost in that case. So that is what telomeres do. They lengthen the DNA, allowing the copying enzyme to complete copying all the meaningful DNA. So you find this question, it is very common, where you asked for the role of telomeres. What are their uses? Number one, we have seen they make DNA longer, such that the copying enzyme is able to copy all the meaningful DNA. And this ensures that when DNA is replicated, the ends of the molecule are all included in replication and not left out, meaning that all the important genes are going to be replicated. and then they'll be passed on to the next daughter cell. Otherwise, if there were no telomeres, the DNA would keep on shortening and daughter cells would miss out on some of the important genetic information that would lead to them not functioning well, and that would lead to their death. So these telomeres are very important in preventing loss of genes during cell division. If the genes are not lost, that means that the cell with all the genes it has received will be able to replicate. It will be able to continuously divide as long as it has all the genetic information. Telomeres have one other use. They will prevent chromosomes from fusing. Like an example, this is our chromosome. You can see here we have many chromosomes inside this cell. So we cannot have this chromosome fusing with this. We cannot have the DNA in this chromosome getting tangled up with this. So telomeres are responsible for preventing ends of chromosomes from fusing. They act as caps. The way you see a cap on a shoelace, that cap on the shoelace protects the shoelace from disintegrating. So telomeres are usually compared with those caps. These telomeres are very important in preventing fusion of chromosomes. So those are the uses of telomeres. And because telomeres are important, all the time when a cell divides, the telomeres shorten. Why? Because remember, we have seen that the enzyme responsible for replicating DNA usually copies the information, but it stops somewhere along the length of the DNA molecule, meaning not all the information is going to be copied. So it stops somewhere. And that is why telomeres keep on shortening. So let's look at this cell. Assuming this cell is going to divide, they are showing you its chromosome with all the telomeres. After the first division, the telomeres shorten because the DNA polymerase stops somewhere. It does not copy the whole DNA molecule up to the end. After the second cell division, you can see the telomeres still are going to shorten. If cell division further goes on, they are really going to shorten so much. Next thing we shall be having is loss of the important genetic information because the whole telomere has shortened, and now what will happen next will be the important genetic information being lost. So there is an enzyme known as telomerase. This enzyme is going to be responsible for topping up the telomeres. So every time a cell divides and the telomeres shorten, that enzyme is responsible for topping up those telomeres. So that is why we look at that enzyme telomerase. It's responsible for maintaining the telomeres. What does it do? It keeps on adding extra bases to that chromosome during each cell cycle. So the moment the cell divides to those daughter cells' DNA, the telomeres are topped up. So this ensures that no vital information will be lost, and that means that that cell will have all the important genetic information and it will be able to continuously divide. So it allows the telomerase enzyme allows cells to have a high number of cell cycles because it can divide and divide multiple times. Like if you look at this cell, if its telomeres were being topped up, it would be able to divide as many times as possible. But now, because the telomeres were not being topped up, it's going to stop dividing. This means that these cells might not divide again because they will be passing on less genetic information as expected. So telomeres are topped up by enzyme telomerase. So that allows a greater ability to replace damaged cells. So if your cells have enzyme telomerase, that means that their telomeres will always be topped up, and that means in case some cells are damaged, then we shall have those cells dividing multiple times to replace those damaged cells. But if someone lacks that enzyme, if their telomeres are not being topped up, that means even if you get a tissue that is damaged, you'll have issues or problems with that tissue or those cells of the tissue being replaced. So Here they are telling us that some cells do not top up their telomeres at each division. So there are some cells that top up, there are those which don't top up. Now, the ones that don't top up are the ones that have differentiated. Like you find we have a red blood cell. It has taken up the role to carry oxygen. So there is no need for it to top up its telomeres. Why? It is not going to divide. But some other cells that continue dividing, but they don't top up their telomeres, time comes and they lose important genetic information. They lose vital DNA. They lose important genes. So as a result, the cells are going to die. And this is one of the mechanisms they think brings about aging. Like you see how people grow old and maybe the skin becomes sagging. It's no longer strong and firm as it was when you were still young. Maybe because some of the cells in that region have divided so many times that they have started losing important genetic information. So eventually, they are not going to be repaired. Eventually, you're going to have a sagging skin. You will not have maybe your collagen made because the cells probably responsible for making collagen, which makes your skin firm, are no longer able to do that role because they have divided multiple times. Their telomeres were not topped up and they have lost important genetic information. So scientists believe that by preventing the loss of telomeres, maybe they will be able to slow down aging and eventually slow down death. So let's look at stem cells. You've heard about stem cells. Stem cells are even used in treatment of different diseases. But what is a stem cell? A stem cell is a cell. It's first of all unspecialized. Unspecialized means that it's not taking up a particular role within your body. Its role is to just divide. So it's an unspecialized cells that divides by mitosis. Like here they are showing us we have one cell. We are referring to it as a stem cell. Why? Because it's able to divide and form other cells. Now, some of the cells that have been formed can decide to specialize and take up special roles. Or they can remain stem cells and just continue dividing. So that is what a stem cell is. Its role is to simply divide by mitosis, giving rise to other cells. Now, these stem cells, when you look at them, they have different powers as far as division is concerned. So the power of a stem cell to produce different types of cell is variable and is referred to as its potency. We could look at this like an example at home. At home, we have a family head. That family head can determine whatever is going to happen within your home. Maybe it's your father. So your father has the highest power, the highest say at home. Then we have your mother. We can have your auntie. We can have you. The power you have is not the same as the power your father has. So even these stem cells, they have different powers. But for us, when we are looking at the powers of stem cells, we are looking at the ability of them to form different types of cells. When they divide, are they able to form different types of cells? So that is what we look at as potency, the ability of a stem cell to produce different types of cells. And as far as potency is concerned, we are going to look at three types of stem cells. So we are going to start with the first type, the totipotent. So totipotent, these are the stem cells with the highest power, if I may put that in quotes, the highest power. They have the total potential to form any type of cell, any type of cell. And this is where we find the zygote. The zygote is a single cell, but the zygote is able to form all the different cells that are going to make up a human being. So the zygote is totipotent. Why? It has the capability of forming all the different types of cells. Your heart will come from the zygote, the placenta, the lungs, and so on and so forth. All of them come from that one cell. So it has the highest potency. You can remember totipotent with maybe total potential. It has that total potential to form any type of cell. And that is where life comes from. So the zygote is totipotent. The zygote, remember, once it has been made, it starts dividing to form from 1 cell to 2 cells, from 2 to 4, from 4 to 8, from 8 to 16. So up to the 16 cell stage, all those cells are totipotent. That is how even identical twins are made. So as long as the zygote has divided up to the stage cells, first cell stage, 8 cell stage, or 16 cell stage, any of those cells, if it breaks off, it can also still have the ability to form a whole human being. So that is how identical twins are formed. You'll find that some of the cells break off and then they develop into another organism. And you know that identical twins are genetically identical. They have the same genes. So those are coming from totipotent cells. So we have stem cells that are totipotent. An example, the zygote. We also have, after totipotent, the next is the pluripotent. Pluripotent, it can form, yes, very many cells, but its potential is not the same as that of the totipotent. Like an example, after the 16 cell stage, the embryo, it can be able to form very many other cells. It can form maybe the nerve cells, the heart muscle cells, but it cannot form another human being. So that is pluripotent. They can form all the cells that lead to the development of the embryo and then later the fetus, but they cannot form a human being or a whole human or a whole organism. So those are pluripotent. Now, once a baby is born, within your body, or even you as an adult within your body, you have some cells that are multipotent. They still have the ability to divide and form different stem cells, but the types they can make are really very few. Like an example of multipotent are the cells in your bone marrow. The cells in your bone marrow are able to divide to form blood cells, but they only form blood cells. They can only form the white blood cells, the red blood cells, lymphocytes, but they cannot form any other type of cell apart from blood cells. So we call that multipotent. They are only able to produce a few types of cells. So that is potency as far as stem cells are concerned. So within an adult, many of your cells do not have the ability to divide. But it is very essential that in different parts of your body, we have some cells remain there. Why? Because anytime your organs, your tissues can be damaged and they need to be repaired. How will they be repaired? By those stem cells which remain around. They'll be producing new cells. Like look at your skin. Your skin can be damaged maybe as you do different activities, but all the time you damage it, it repairs and grows back. Why? Because underneath your skin, there are stem cells responsible for producing new skin cells. Within your gut, as you eat food, as digestion occurs, cells are damaged. So your gut has stem cells that are responsible for always dividing to replace the ones that have been damaged. Your brain has stem cells. We have already looked at your bone marrow, which has stem cells that are multipotent, that can form the different types of blood cells, the red blood cells, the monocyte, the neutrophils, the lymphocytes. You look at the roles of these cells as you learn about immunity. So those are stem cells. And stem cells can be used to treat different diseases. That takes us to stem cell therapy. So here we are looking at the introduction of a new adult stem cell into a damaged tissue to treat a disease. An example, you've heard about people that have sickle cell anemia. People with sickle cell anemia have their bone marrow producing faulty red blood cells. That means that their bone marrow has those stem cells, but the stem cells make faulty red blood cells. So what is the solution? The solution is, if this person has a bone marrow whose stem cells release faulty or make faulty red blood cells, why can't we remove those faulty stem cells from their bone marrow and we get the bone marrow of a normal person whose stem cells release or make the right red blood cell? So that is what bone marrow transplant is about. They remove the faulty stem cells that divide to form faulty red blood cells and give you with normal ones from someone whose bone marrow makes new red blood cells that are good, and they're going to be performing their role very well. Cancer treatment. So with cancer treatment, someone could have cancer in a certain part of the body, that those cancerous cells are going to be killed, maybe using radiations. And after those cells are killed, they can be replaced with new cells that can start dividing and replace those cancer cells that were killed and removed from you. And it is believed that in the future, the stem cell therapy is going to be used to treat very many diseases like diabetes. We know with diabetes, some of the types of diabetes, you can have your cells of the pancreas maybe damaged in that they can no longer secrete insulin. So why not provide you with stem cells that can divide from those cells such that they start secreting insulin again? Muscle and nerve damage. If you have your nerves damaged, can't we have stem cells placed into that part of the body that is affected so that they can divide and form the nerves again? They can even be used, they are projecting that they will be used to treat brain disorders such as Parkinson's and Huntington's disease. Now, these are hereditary diseases. And if we have stem cells, they can be used to treat such diseases. More research is being done. They are also experimenting growing new tissues. If I can isolate these stem cells in a lab and maybe someone needs a heart transplant, can't I grow these stem cells and I make them differentiate to form maybe heart muscle cells or to form liver cells? So that takes us into making new organs in a lab. So that is a branch that you can consider getting into. And I'm sure if you contribute to that research, you'll be making lots and lots of money. So we are having a question here that as a result of mitosis, all 200 plus types of cell contain the same set of genes as the zygote. What does this suggest about the mechanism by which cells become different? So here they are trying to tell you, when you look at all the different cells, an example, the different cells in your body, they all have the same genes. Look at the cells in your eyes, the cells in your tongue, the cells in your nose, the cells in your liver. They all have the same genes. They all came from the same zygote, meaning they are genetically identical. But how come they are different? They are different. They are not the same. When you look at the cones, which are cells in your eyes, they are not the same as the cells in your tongue. They are not the same as the red blood cell. So they're asking you, what does that suggest about the mechanism by which cells become different? Yeah, that is food for thought. Food for thought. I'm leaving it behind. If this video has helped you, please give it a thumbs up. Kindly share, kindly subscribe to my channel. I am going to give you as much information as you need to ensure that you excel. I do not teach students that don't excel. So kindly subscribe, share with your friends, invite your friends to join my channel. See you on the next one. Bye.